Now, while on every single Western blot protocol, we're going to be looking at a specific protein present on the membrane, there can be different applications for that technique. Originally, Western blots were developed so that we could target one specific protein on a membrane and simply know if that protein was present in that sample or not. At that point, Western blots were merely qualitative. That is still one of the most common applications of Western blots. But advancements in the technique, such as the use of a validated internal loading control for normalization, now allow that technique to actually be quantitative, presuming that you have planned, you have executed, and you have taken your time to optimize that protocol very carefully. So then again, if we do all those things, your Western blot can be used to allow you to estimate the relative abundance of proteins in those samples. Not only that, but there have also been advancements in how primary antibodies are generated and selected. Those advancements have been so big that nowadays we can actually target not only a certain protein, but specific modifications on those proteins. So Western blots can actually be used nowadays to detect specific post-translational modifications such as phosphorylation and ubiquitination. Another cool thing that we can do is use a molecular weight marker, also known as a protein ladder. On those molecular weight markers, we have proteins of known molecular weights that are labeled. And when we load those markers together with our test samples, we can actually estimate the size of our proteins. Now, there are many other applications for Western blot, most of them somewhat complex, such as identifying protein-to-protein interaction, or perhaps studying enzymatic activity, or even identifying which cell compartment a specific protein is localized. But we're not going to be talking about those on these series of Lambda U courses. We're going to save those for more advanced Lambda U courses. Now, obviously, as we just talked about, there's several applications for Western blotting. It is a very popular technique that a lot of labs run on a daily basis. So what we're going to do now is we're going to briefly do an overview of the three main parts of the Western blot protocol. We're going to save the details of the science behind each step for the next courses, but let's start talking about the protocol so we can get familiar with it.